Hello everyone. Today we are going to discuss the major receptor families or the type of receptors in our body. We classify receptors into four types of receptors and we are now going to discuss each type. So let's start. First type of receptors are called ligand-gated ion channels. So basically these type of receptors are channels, ion channels, and these ion channels are ligand gated. So now we are comparing this type of ion channels with voltage gated ion channels, which we discussed when we were talking about action potential. And voltage gated ion channels were ion channels that was opened or closed according to the electricity or the voltage. But ligand-gated ion channels is or are ion channels that open when a ligand binds to it. So their gates are opened when a specific ligand binds to it. So in a very simple way, these type of receptors are channels, ion channels, or part of them are the ion channels. Okay, so what are the ligand-gated ion channels in our body? For example, this is a cell at which its surface there are ion channels, and there is a groove here to which our ligand binds. And when our ligand binds to this ion channel or this site at the ion channel, it opens and ions gets away into the cell. Okay, this is very simple. And a very important example of ligand-gated ion channels are the nicotinic cholinergic receptors. These receptors are receptors to which a molecule called acetylcholine binds. These receptors are called nicotinic receptors because their agonist is also nicotine, so they are called nicotinic cholinergic receptors. How these receptors work? These receptors or these ion channels are sodium channels. When a ligand or an agonist binds to it, it opens and sodium enters into the cell, because as we studied before, sodium is found in a larger concentration outside the cell. So it moves according to its concentration gradient from outside the cell to inside the cell. So sodium enters into the cell, there is a sodium influx when acetylcholine or nicotine binds to the nicotinic receptors. So here we found a nerve that innervates skeletal muscle. This nerve is called somatic nerve, and on this nerve on this skeletal muscle, on its cell membrane, we find the nicotinic receptor. And the space between the somatic nerve and the skeletal muscle is called neuromuscular junction. And this receptor, acetylcholine, binds to it. This receptor is ligand-gated ion channel. And when it binds to it, sodium enters the cell. And when sodium enters the cell, any cell, it causes depolarization, as we discussed before. And the depolarization in a skeletal muscle increases calcium, increases intracellular calcium in the cells of the skeletal muscle by allowing its efflux from the sarcoplasmic reticulum. So, again, somatic nerve releases acetylcholine. This acetylcholine binds to the nicotinic cholinergic receptors on skeletal muscle cells. This receptor is called NM. N is for nicotinic and M for muscle. So this is the nicotinic receptor of muscle. Because we would know later on that there is another type of nicotinic receptors, but not on the muscles. Okay. So at the neuromuscular junction, somatic nerve releases acetylcholine that binds to NM receptor, and NM receptor is a ligand-gated ion channel receptor of sodium channel. So it opens and sodium moves according to its concentration gradient from outside the cell to inside the cell, causing depolarization, so increasing calcium level in the skeletal muscle. And what does calcium do? Yes, it causes contraction of the skeletal muscle. So the action of acetylcholine on NM receptor at neuromuscular junction causes contraction of skeletal muscles. Okay, and this is an important example of a ligand-gated ion channel receptor. So let's see another type of ligand-gated ion channel, which is the gamma amino butyric acid receptors, or for short GABA receptors. Gamma amino butyric acid is an inhibitory neurotransmitter in central nervous system. And why GABA is an inhibitory neurotransmitter? Because when GABA, which is the agonist of these receptors, binds to it, it causes the influx of chloride inside the cell. Chloride is negatively charged ion, which presents in larger amount outside the cell. So these GABA receptors are ligand-gated ion channels that opens when GABA binds to it. Then when they open, there is chloride influx inside the cell. So this results in more negative inside the cell. And initially at resting state, when cell is in polarized state, as we learned before, there is more negative inside the cell. So when we are adding more negative by the influx of chloride, we are causing hyperpolarization. Hyperpolarization. It is more negative inside the cell. Instead of starting with negative 70 millivolt, which we discussed before, we have more negative. We have around, we can say, negative 80 millivolt. And do you remember the threshold level? Yes, it was around negative 55 millivolt. And we learned that when we hit or reach this threshold, there is a sudden and enormous opening of many and many sodium channels, and there is depolarization occurs. So if we are now having hyperpolarization, we are now more far from this threshold level. We are now more far from this level. So our cell couldn't have an action easily. It needs a larger stimulus to reach a threshold level and having an action. So by this mechanism, we have inhibition of the cell. Our cell is somehow inhibited by this hyperpolarization. So for ligand-gated ion channels or type 1 receptor family, we have nicotinic cholinergic receptors, which are sodium channels, and we have gamma aminobutyric acid or GABA receptors as chloride channels.